Okay, we say today we go to clinical bacteriology. So clinical means truly some bacteria cause illness, and we need to study, and then what they look like and how we do some preventing and against them. So before we start, we talking about ginseng first. What means ginseng? So that's the way we identify for bacteria. Generally speaking, we separate bacteria with two big categories. So we're talking about by the shape, we separate with three categories before. Now we're talking about with ginseng, we separate with two big categories. So one category we're talking about positive. One category we're talking about negative. So by this way, we identify with different kind of bacteria. And the different kind of bacteria, we can use different kind of antibiotics to kill them. Yeah. So that's the first step. We try to separate or identify with ginseng. So let's still go to our video first.
Gram stain. Gram stain or gram staining, also called Gram's method, is a method of staining used to distinguish and classify bacterial species into two large groups, gram-positive bacteria and gram-negative bacteria. The name comes from the Danish bacteriologist Hans Christian Gram, who developed the technique. Gram staining differentiates bacteria by the chemical and physical properties of their cell walls. Gram-positive cells have a thick layer of peptidoglycan in the cell wall that retains the primary stain, crystal violet. Gram-negative cells have a thinner peptidoglycan layer that allows the crystal violet to wash out on addition of ethanol. They are stained pink or red by the counterstain, commonly safranine or fuchsin. Lugol's iodine solution is always added after addition of crystal violet to strengthen the bonds of the stain with the cell membrane. Gram staining is almost always the first step in the preliminary identification of a bacterial organism. While gram staining is a valuable diagnostic tool in both clinical and research settings, not all bacteria can be definitively classified by this technique. This gives rise to gram-variable and gram-indeterminate groups. The method is named after its inventor, the Danish scientist Hans Christian Gram, 1853 to 1938, who developed the technique while working with Karl Friedlander in the morgue of the City Hospital in Berlin in 1884. Gram devised his technique not for the purpose of distinguishing one type of bacterium from another but to make bacteria more visible in stained sections of lung tissue. He published his method in 1884, and included in his short report the observation that the typhus bacillus did not retain the stain. Gram staining is a bacteriological laboratory technique used to differentiate bacterial species into two large groups, gram-positive and gram-negative, based on the physical properties of their cell walls. The gram stain is not an infallible tool for diagnosis, identification, or phylogeny, and it is of extremely limited use in environmental microbiology. It is used mainly to make a preliminary morphologic identification or to establish that there are significant numbers of bacteria in a clinical specimen. It cannot identify bacteria to the species level, and for most medical conditions, it should not be used as the sole method of bacterial identification. In clinical microbiology laboratories, it is used in combination with other traditional and molecular techniques to identify bacteria. Some organisms are gram-variable, meaning they may stain either negative or positive. Some are not stained with either dye used in the gram technique and are not seen. In a modern environmental or molecular microbiology lab, most identification is done using genetic sequences and other molecular techniques, which are far more specific and informative than differential staining. Gram stains are performed on body fluid or biopsy when infection is suspected. Gram stains yield results much more quickly than culturing, and are especially important when infection would make an important difference in the patient's treatment and prognosis. Examples are cerebrospinal fluid for meningitis and synovial fluid for septic arthritis. Staining mechanism. Purple stained gram-positive and pink stained gram-negative. Gram-positive bacteria have a thick mesh-like cell wall made of peptidoglycan, 50 to 90% of cell envelope, and as a result are stained purple by crystal violet, whereas gram-negative bacteria have a thinner layer, 10% of cell envelope. So do not retain the purple stain and are counterstained pink by safranine. There are 4 basic steps of the gram stain. Applying a primary stain, crystal violet, to a heat-fixed smear of a bacterial culture. Heat fixation kills some bacteria but is mostly used to affix the bacteria to the slides so that they don't rinse out during the staining procedure. The addition of iodide, which binds to crystal violet and traps it in the cell. Rapid decolorization with ethanol or acetone. Counterstaining with safranine. Carbolfuchsin is sometimes substituted for safranine since it more intensely stains anaerobic bacteria, but it is less commonly used as a counterstain. Crystal violet, CV, dissociates in aqueous solutions into CV and zinc chloride, Cl, ions. These ions penetrate the cell wall of both gram-positive and gram-negative cells. The CV and ion interacts with negatively charged components of bacterial cells and stains the cells purple. Iodide interacts with CV and forms large complexes of crystal violet and iodine, CV, I, within the inner and outer layers of the cell. Iodine is often referred to as a mordant, but is a trapping agent that prevents the removal of the CV, I complex and, therefore, colors the cell. When a decolorizer such as alcohol or acetone is added, it interacts with the lipids of the cell membrane. A gram-negative cell loses its outer lipopolysaccharide membrane, and the inner peptidoglycan layer is left exposed. The CV, I complexes are washed from the gram-negative cell along with the outer membrane. In contrast, a gram-positive cell becomes dehydrated from an ethanol treatment. The large CV, I complexes become trapped within the gram-positive cell due to the multilayered nature of its peptidoglycan. The decolorization step is critical and must be timed correctly. The crystal violet stain is removed from both gram-positive and negative cells if the decolorizing agent is left on too long, a matter of seconds. After decolorization, the gram-positive cell remains purple and the gram-negative cell loses its purple color. Counterstain, which is usually positively charged safranine or basic fuchsin, is applied last to give decolorized gram-negative bacteria a pink or red color. Both gram-positive bacteria and gram-negative bacteria pick up the counterstain. The counterstain, however, is unseen on gram-positive bacteria because of the darker crystal violet stain. Gram-positive bacteria. Gram-positive bacteria generally have a single membrane, monoderm, surrounded by a thick peptidoglycan. Some bacteria have cell walls which are particularly adept at retaining stains. These will appear positive by gram stain even though they are not closely related to other gram-positive bacteria. These are called acid-fast bacteria, and can only be differentiated from other gram-positive bacteria by special staining procedures. Gram-negative bacteria. Gram-negative bacteria generally possess a thin layer of peptidoglycan between two membranes. Most bacterial phyla are gram-negative. Gram-variable and gram-indeterminate bacteria. Some bacteria, after staining with a gram stain, yield a gram-variable pattern. A mix of pink and purple cells are seen. Gram-indeterminate bacteria do not respond predictably to gram staining and, therefore, cannot be determined as either gram-positive or gram-negative. The term gram staining is derived from the surname of Hans Christian Gram. The eponym, gram, is therefore capitalized but not the common noun, stain, as is usual for scientific terms. The initial letters of gram-positive and gram-negative, which are eponymous adjectives, can be either capital G or lowercase g, depending on what style guide, if any, governs the document being written.
Which is called Grimstein, yeah. So that's the way, we say we can separate bacteria with two big groups. So one group is positive, another group is negative. So the positive kind of group after stain is blue color. After stain is blue color. And the negative kind of group after green stain, is the red color. So that's we say we separate with two groups. So that's the mainly way we identify for many kind of bacteria. And we can choose antibiotics. So because negative kind of bacteria and positive kind of bacteria, we can use different antibiotics. So that's under microscope, some bacteria look like. So you can see some bacteria is blue color, like the group here. That's green stem positive kind of bacteria. And some bacteria is red color here, is green stem negative kind of bacteria. So that's certainly these two bacteria, the shape is different also. But just let us to compare different kind of bacteria. So gram-positive kind of bacteria, we still have some rod shape in there, like we talked about before. And they still possible have some long shape in there, like we talked about before. So that's go to different kind of bacteria. Then we talking about some gram-negative kind of bacteria. Gram-negative kind of bacteria, we still have the similar shape in there with gram-positive. And we also have some special kind of bacteria we talked about last time. They may have some benefits there. So that's we name
Mycobacteria.
it. Mycobacteria.
Mycobacteria.
So some bacteria's name, they coming from Latin words. They may be a little bit strange in there, and maybe a little bit long words there. So I just put some standing pronunciation there for you easily like remember and review. So like each of the words, try to speak twice here.
Mycobacteria. Mycobacteria.
And some of the gram-positive kind of bacteria, they like a lot of lines in there. We see the pictures later, but we name it
Actinomycetes. Actinomycetes.
So this part is basically we talking about gram-positive, gram-positive. And even we say it's gram-positive, but still some of them not that sensitive to gram stem. We're talking about some detail later, but generally say with this group here belong to gram positive. Then gram negative. Gram negative is a big category. It's more commonly in clinically and cause illness. So you can see this side, we still have round shape there. We still have like a long shape there.
Bacilli. Bacilli.
And also this side, also this side still belong to gram negative. They including
Mycoplasmas. Mycoplasmas.
and
Spirochetes. Spirochetes.
and
Chlamydia. Chlamydia.
and another kind.
Rickettsii. Rickettsii.
So that's basically this special kind still belong to gram negative. We talk about some detail later. And also we say long kind of shape of gram negative kind of bacteria. We still have many kind of different kind. So basically clinically, by they cause the illness, by they cause the illness, we separate with some group here. Like if they cause the intestine infections, intestines, digestion systems, intestine infections, we pick one group there. And some group is more relationship with reproduction systems infections. We put it for the second group. And then some illness caused by bacteria, they transform like by some animals. We put for another group here. So that's mainly we like separate bacteria by different group. The first big category, gram positive and gram negative. And then we have different kind. So let's talking about it one by one. So we say the first category, we're talking about gram positive
Cocci.
and
Cocci.
round shape, gram positive and round shape, round shape. We call cocci, that's last week we talked about cocci. And in this category, basically clinically pretty commonly we have two small categories here, two small categories here.
Staphylococcus. Staphylococcus.
So one category we call it Staphylococcus, means they are grouped together like a group. Like group. We still talk about some detail later. Another small category, they look like connecting each other like a lane in there.
Streptococcus. Streptococcus.
That's we name it. Streptococcus. Both round shape. Both round shape. We say both of them belong to gram positive cocci. Gram positive cocci. But there are some different shape in there. And they also both producing special kind of enzyme.
Catalase. Catalase.
So that's basically we're talking about. Oh, the first one producing that special kind of catalase.
Catalase.
Yeah. The second one didn't have. So that's we separate. Yeah, that's we separate. So that's the shape here. Yeah, that's the shape here. You can see that's round shape and group together, look like they are group together. They are both the same.
Staphylococcus.
Yeah.
Staphylococcus.
They are the same bacteria. Just when we use the microscope, one is more time, like this one is more times. Yeah, it looks bigger, but they are the same. Okay, now we're talking about the bacterial relationship with the illness.
Staphylococcus. Staphylococcus.
Oh, I try to likely connect it. Yes, Staphylococcus means we say that's a rock shape, like a group of bacteria together. So like this picture here. Does they not moving again?
我就是看到那个角。
Staphylococcus.
Yeah, look like some.
Staphylococcus.
So this one is a specialist Staphylococcus. Is bacterial gram positive. Yeah, gram positive. In this kind of category, we still have different kind of bacteria. So one bacteria one kind of bacteria, they cause the different kinds of infections and can cause severe kind of infections. Like the picture here, I think you see the picture here is a swollen fig there. And that's basically caused by this kind of bacteria. So that's look like.
Staphylococcus. Staphylococcus.
So these bacteria, they cause a lot of illness. Yeah, they cause a lot of illness. And clinically, pretty commonly, we can see these bacterial infections. So we still have a radio for this illness.
Staphylococcus aureus. Staphylo means grape shape. Coccus means round. Aureus means golden color. Staphylococcus aureus is a gram-positive, round-shaped bacterium, and it is a member of the common microbiota of the human body. Frequently found in the upper respiratory tract and on the skin. It is a facultative anaerobe that can grow without the need for oxygen. Although Staphylococcus aureus usually acts as a commensal of the human microbiota, it can also become an opportunistic pathogen, being a common cause of skin infections, including abscesses, respiratory infections such as sinusitis, and food poisoning. Pathogenic strains often promote infections by producing virulence factors. such as potent protein toxins, and the expression of a cell surface protein that binds and inactivates antibodies. The emergence of antibiotic-resistant Staphylococcus aureus, MRSA, is a worldwide clinical problem. Despite much research and development, no vaccine for Staphylococcus aureus has been approved. An estimated 20% to 30% of the human population are long-term carriers Staphylococcus aureus, which can be found as part of the normal skin flora, in the nostrils, and as a normal inhabitant of the lower reproductive tract of women. Staphylococcus aureus can cause a range of illnesses, from minor skin infections, such as pimples, impetigo, cellulitis, folliculitis, carbuncles, scalded skin syndrome, and abscesses, to life-threatening diseases, such as pneumonia, meningitis, osteomyelitis, endocarditis, toxic shock syndrome, bacteremia, and sepsis. It is still one of the 5 most common causes of hospital-acquired infections, and is often the cause of wound infections following surgery. Each year, around 500,000 patients in hospitals of the United States contract a staphylococcal infection, chiefly by Staphylococcus aureus. Up to 50,000 deaths each year in the USA are linked with Staphylococcus aureus infections. 1881, Sir Alexander Ogston, a Scottish surgeon, discovered that Staphylococcus can cause wound infections, after noticing groups of bacteria in pus from a surgical abscess during a procedure he was performing. He named it Staphylococcus, after its clustered appearance evident under a microscope. Then, in 1884, German scientist Friedrich Julius Rosenbach identified Staphylococcus aureus, discriminating and separating it from Staphylococcus albus, a related bacterium. In the early 1930s, doctors began to use a more streamlined test to detect the presence of a Staphylococcus aureus infection by the means of coagulase testing, which enables detection of an enzyme produced by the bacterium. Prior to the 1940s, Staphylococcus aureus infections were fatal in the majority of patients. However, doctors discovered that the use of penicillin could cure Staphylococcus aureus infections. Unfortunately, by the end of the 1940s, penicillin resistance became widespread amongst this bacterium population, and outbreaks of the resistant strain began to occur. Staphylococcus aureus can be sorted into 10 dominant human lineages. There are numerous minor lineages as well, but these are not seen in the population as often. Only a few strains of Staphylococcus aureus are associated with infections in humans. Another notable evolutionary process within the Staphylococcus aureus species is its coevolution with its human hosts. Over time, this parasitic relationship has led to the bacterium's ability to be carried in the nasopharynx of humans without causing symptoms or infection. This allows it to be passed throughout the human population, increasing its fitness as a species. However, only approximately 50% of the human population are carriers of Staphylococcus aureus, with 20% as continuous carriers and 30% as intermittent. This leads scientists to believe that there are many factors that determine whether Staphylococcus aureus is carried asymptomatically in humans, including factors that are specific to an individual person. According to a 1995 study by Hoffman et al., these factors may include age, sex, diabetes, and smoking. They also determine some genetic variations in humans that lead to an increased ability for Staphylococcus aureus to colonize. In conclusion, there is evidence that any strain of this bacterium can become invasive as this is highly dependent upon human factors. Microbiology. Key characteristics of Staphylococcus aureus. Staphylococcus aureus. Grape cluster berry. Aureus, golden. It is a facultative anaerobic. Gram-positive cockle, round. Bacterium also known as golden staph. Staphylococcus aureus is non-motile and does not form spores. In medical literature, the bacterium is often referred to as Staph aureus. Staphylococcus aureus appears as staphylococci, grape-like clusters, when viewed through a microscope, and has large, round, golden yellow colonies, often with hemolysis, when grown on blood agar plates. Staphylococcus aureus reproduces asexually by binary fission. Complete separation of the daughter cells is mediated by Staphylococcus aureus autolysin, and in its absence or targeted inhibition, the daughter cells remain attached to one another and appear as clusters. Staphylococcus aureus is catalase positive, meaning it can produce the enzyme catalase. Catalase converts hydrogen peroxide to water and oxygen. Catalase activity tests are sometimes used to distinguish staphylococci from enterococci and streptococci. This is the end about Staphylococcus aureus.
So these bacteria, these bacteria usually they cause some infection in the salt air, like the picture here. So usually they will not cause that kind of severe, like the last bacteria we're talking about. And we also have some other kind of bacteria, and they cause other illness. Yeah, I still need to plan here, otherwise they will no sounds here. Yeah, maybe we go to there first and then we come back.
Neisseria meningitidis. Neisseria meningitidis. Neisseria gonorrhoeae. Neisseria gonorrhoeae. Meningococcemia. Meningococcemia. Gonorrhea. Gonorrhea. Neonatal conjunctivitis. Neonatal conjunctivitis.
So that's some illness name. Yeah, that's some illness name. We are talking about gram-negative kind of bacteria, and they have still subcategory there. Yeah, subcategory there.
The penicillin doesn't work against this kind?
Not really. Yeah, not really. Usually
okay.
penicillin working with gram-positive. Yes.
Right, okay.
Yeah.
How do they kill this kind?
We have another different kind of antibiotics. Yeah.
Okay.
Yeah, but generally for bacteria, we have effective antibiotics to against them, but not for all the viruses. So we're talking about bacteria. Yeah, we're talking about bacteria here. We say it causes meningitis. Meningitis is the infection on the cover of the brain. Yeah, so that's we say that's called meningitis. And maybe also go to like a covered spinal cord. That's we talked about before. Infection in there. Yeah, infection in there. And another illness here belong to sex illness. Yeah, sex illness. Pretty similar like HIV, but not that severe. And HIV, we say that's caused by virus, but this one caused by bacteria. Yeah. And another illness here that's especially for new baby got conjunctivitis by this kind of bacteria. So these kind of bacteria still cause a lot of illness. Yeah, so that's for gram-negative kind of bacteria. We have different kind. Yeah, we have different kind. Some bacteria they especially cause the meningitis, and some bacteria they especially cause like sex kind of illness, and that's still the same thing here. That's still this picture here. Yeah, show you what happens there. And some of them maybe cause new babies conjunctivitis. That means happens outside of the eyeball, conjunctival area, and even the eye leaders there. Some infection and swollen in there. You see the new baby even couldn't open the eyes. And then we're talking about gram-positive kind of long kind of shape of the bacteria. So these bacteria we're talking about, they have different kind of shape. Yeah. Some of them need oxygen. Some of them don't need oxygen. So we still have different like the shape, and some of them can form the spores. Yeah. And some of them don't form the spores. Yeah. So that's cause different illness. So that's belong to gram-positive. Yeah. Still belong to gram-positive rod. Yeah. And we also have some gram-positive rod bacteria. They are not formed spores. Yeah, not formed spores. And certainly they cause different illness. Like some bacteria, they can especially cause the illness on the salt air and form a like a white kind of shape cover there. Yeah, this kind of not full script here. I couldn't make notes here, but I read in here. Yeah, read in here. And then by the bacterial shape, and especially sometimes by like how bigger the nuclear air, or maybe the single nuclear air, we name the different bacteria. So that they still cause the different illness, still can cause meningitis. So even different bacteria, maybe they cause the similar illness in similar part of the body. And the same bacteria will possible cause the illness in different part of the body. So that's we're talking about the bacteria. And I like to say a special kind of bacteria, E. coli. Yeah, E. coli. So E. coli is a pretty common kind of bacteria. It usually in our large intestine is normally living there. We can thinking about the normal flora. Yeah, normal flora. But this bacteria is go to other part of our body. That will be cause the other troubles. That will cause the illness. Like mostly they will go to urination tract, including your bladder or maybe kidney. So that can cause the infection. That's a pretty common one. Yeah, pretty common one. It belong to gram-negative kind of rot. Yeah, gram-negative rot. And certainly they also cause the other illness. For new babies, still can cause the meningitis. And maybe sometimes they even cause the diarrhea. And then we're talking about gram-negative kind of rot, kind of bacteria. We separate by they cause the illness. They can cause respiration tract infections. We have different kind of bacteria there. Mainly is 3 in there. And I got some detail here. Just give you some basic, some idea what kind of illness they will cause. And we also have some gram-negative kind of rot. Its relationship with some animals. Yeah, like animal can transform this kind of bacteria to humans. So that's we have. And then we're talking about some bacteria, like a bunch kind of shape in there. And that kind of bacteria, we say, still belong to gram-positive. But they are not that easily to dying use gram stem. Yeah, even they belong to gram-positive. They still blue color, yeah, blue color. So that's we have some of them there. And another small category. So like you can see the picture shape there. They still similar like that one. They still belong to gram-positive, but still not that easily got a gram stem. And then we have more belong to gram-negative. Yeah, gram-negative. So that's another group of small category there. So usually they don't have big sink well in there. Yeah. Just small kind of cell well in there. So that's why we put for another category. And finally, we go to some bacteria, the special kind of spiroid. It usually this here belong to gram-negative. So we have different kind and cause different illness. And another small group, gram-negative. So the shape, I just put some picture for your easily understanding. So these kind of bacteria usually easily cause the urination tract infections and maybe for vagina infections in there normally. And certainly can cause the other infections like lung infection, pneumonia, or maybe some infection in the eyes. And finally, we go to another subcategory here, the subcategory there. So belong to gram-negative, still belong to gram-negative. And they also cause a special kind of illness. So that's basically we're talking about clinically. Bacteria can cause the illness. You may not that easily to remember all the bacteria's name, but at least you should remember what means gram stem, and what's the color of the gram stem, and the category, like which category belong to gram positive, and which category or subcategory belong to gram-negative. So that's for today's class.